Good afternoon, and welcome to the Brain and Behavior Research Foundation's Meet the Scientists monthly webinar series. I'm Dr. Jeff Borenstein, President and CEO of the Foundation, and your host for today's webinar. Today, Dr. Christopher Lowry will present the microbiome and mental health. The Brain and Behavior Research Foundation is committed to alleviating the suffering caused by mental illness by awarding grants that will lead to advances and breakthroughs in scientific research. The Foundation is the largest private funder of mental health research grants. Since 1987, the Foundation has awarded more than $365 million to fund more than 5,000 grants to over 4,000 scientists around the world. 100% of all donor contributions for research are invested in our grants to scientists who are working to find breakthroughs in disorders such as addiction, ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, OCD, PTSD, and schizophrenia. I'm delighted to introduce Dr. Christopher Lowry. Dr. Lowry is Associate Professor in the Department of Integrative Physiology and Center for Neuroscience at the University of Colorado, Boulder. Dr. Lowry is also the recipient of a 2007 and 2010 Foundation Young Investigator Grant. Today's webinar will begin with his presentation. This will be followed by a question and answer period. To submit your questions, please use the Questions tab on the control panel on your screen. Feel free to submit your questions throughout the presentation, but when the presentation is finished, I'll present your questions and will address as many as possible in the time allotted. And now, I'm pleased to introduce Dr. Christopher Lowry. Chris, the floor is yours.
Thank you very much. Thank you, Dr. Borenstein, for the introduction. Thank you to all the attendees of the webinar, and especially thank you to the Brain and Behavior Research Foundation for all the work that you're doing to support research to advance mental health, treatment of mental health. So let's get started. Thank you for attending. Would like to disclose I'm a member of the Scientific Advisory Board of Immunoregulation Therapeutics, which is a company in London that is interested in use of microbiome-based products, specifically mycobacteria. Okay, so the outline for today's talk, I'll start with the idea of prevention of mental health disorders. I'll then go on and introduce you to what's considered the hygiene hypothesis and the possibility that the hygiene hypothesis may be linked in some way to psychiatric disorders. Third, I'll talk about psychiatric disorders as disorders of a failure of immunoregulation. So this may be an unfamiliar term, immunoregulation, but think of it as the body's own mechanisms for suppressing inflammation. So inflammation can be a good thing when you have an infection, but inappropriate inflammation is not a good thing, as we understand from cardiovascular disease. And we'll also see that inflammation may be playing a role in psychiatric disorders as well. And then I'll present some data from some research that we've done suggesting that restoring immunoregulation in individuals that have impaired immunoregulation can prevent development of a PTSD-like syndrome in some animal models. I'll then talk about some future research, including some clinical trials or clinical studies that we're doing in PTSD subjects. So first, the idea of prevention of mental health disorders. This is a quote from a paper by Tom Insel, who was then the director of the National Institute of Mental Health. And over 10 years ago, he and his colleagues raised the idea that in contrast to researchers in cancer and heart disease who've sought cures and preventions, biological psychiatrists in both academia and industry have set their sights on incremental and marketable advances such as drugs with fewer adverse effects. I'd like to point out that major depression is common and becoming more common. So in this paper in Science from 1996, unipolar major depression was ranked number 4 in terms of disability-adjusted life years worldwide and was to be number 2 by the year 2020, ranked only second to ischemic heart disease. Back to Dr. Insel and his idea of preventing mental health disorders. They went on to say that psychiatry will need to develop strategies for prevention of each of these disorders, and in that context they were talking about schizophrenia, mood disorders, and autism. So if we think about the idea of preventing mental health disorders, where should we start? And it's not a particularly straightforward question, but we'd argue that we should think about the risk factors for psychiatric disorders. Some of these are well known, including a genetic predisposition, so the DNA that you inherit from your parents has an influence on your risk for psychiatric disorders. There are also environmental influences, and these come in many forms, including adverse childhood experiences, which you may have heard of as ACEs. But we'd also argue that microbial inputs, particularly during early life, may be important environmental influences. And here we're talking about the microbiome. And so the microbiome is essentially the collection of microorganisms that live on and in the human body. And one of the most widely populated parts of the body is our gastrointestinal tract, particularly our large intestine or colon. And so there can be, on the one hand, pro-inflammatory microbial inputs. So these include bacteria or viruses that can cause disease and inflammation. But on the other hand, the side that we don't think of as frequently is a group of microbial inputs that we would consider immunoregulatory. So if pro-inflammatory microbial inputs is the yin, then immunoregulatory microbial inputs are the yang. And their role seems to be suppressing inappropriate levels of inflammation that would be disadvantageous to the host, and in some cases to the microbes themselves. And so this lack of microbial inputs can lead to a failure of immunoregulation. We'll talk about this in more detail later. And without adequate immunoregulation, we see increases in inflammation. And as we'll see in modern urban societies like the United States and European countries, there's evidence for chronic low-grade inflammation, which is thought to increase our risk for psychiatric disorders. So let's turn our attention to the hygiene hypothesis in psychiatric disorders. This is a paper that we published in 2008, nearly 10 years ago, together with my colleague Graham Rook, who's played a prominent role in evaluating the hygiene hypothesis. And here's a quote from the paper: "Some psychiatric disorders in developed countries might be attributable to failure of immunoregulatory circuits to terminate ongoing inflammatory responses." So how does that really work in terms of our immune system? At the top of this photo that you see on your screen now is what are called the old friends. We'll talk about what types of microorganisms are included among the old friends, but essentially these are organisms that have co-evolved with humans and have adopted this role of suppressing appropriate inflammation. They can do that by binding to immune cells, including immature dendritic cells, which are labeled DC on the figure. And this drives differentiation of these dendritic cells into what are considered regulatory dendritic cells. And regulatory dendritic cells are particularly adept at inducing the differentiation of T cells toward what are called regulatory T cells, or Treg. You can see down in the lower right-hand or lower left-hand corner of the photo. So if I use my pointer, here's the Treg. And these regulatory T cells secrete large amounts of interleukin-10 and TGF-beta. And these are molecules that are secreted by Treg cells that have anti-inflammatory effects. And as such, they can suppress inappropriate inflammation in response to self, inappropriate inflammation in response to gut contents, and inappropriate inflammation in response to allergens. On the other hand, just having IL-10 and TGF-beta around has a general overall anti-inflammatory bystander suppression of inflammation. So here's another way of looking at it. Our ancestral condition was hunter-gatherer societies, small groups of humans living together, eventually engaged in agriculture and maintaining domestic animals, living near and interacting with these animals. And these animals and the lifestyle associated with hunter-gatherer and agricultural societies resulted in adequate levels of interleukin-10 and TGF-beta, these anti-inflammatory cytokines, which can terminate inappropriate inflammation, and that's associated with the normal ratio of these regulatory to effector T cells. So when we say effector T cells, these are the ones that are driving inflammation. There can be different types of inflammation, which are referred to here as Th1 or Th2 type inflammation that are driven by different types of pro-inflammatory molecules. But what we find is that in modern urban societies, we lack the exposure to these types of microorganisms, particularly during early life, including the first 2 years of life. And this results in a low ratio of regulatory to effector T cells. Consequently, this results in increases in inflammation, which has been associated with depression, multiple sclerosis, type 1 diabetes, and Crohn's disease. On the other hand, Th2 type inflammation has been associated with anxiety and allergies. It's worth noting that there's been a very rapid increase in inflammatory conditions in modern urban societies since about the 1950s to 1960s. And so this is a graph from a paper in 2002 in the New England Journal of Medicine showing that the incidence of immune disorders has been increasing dramatically since that time, and this includes multiple sclerosis, Crohn's disease, which is a type of inflammatory bowel disease, type 1 diabetes, and autoimmune disease, and asthma. And early versions of the hygiene hypothesis interpreted these immunological changes in urban societies as associated with a shift toward a Th2 type immune response. But it's now clear that essentially all types of inflammation are increasing in modern urban societies, including Th1 and Th17 dominated immune disorders. There has been some limited work comparing biomarkers of inflammation in rural settings like lowland Ecuador compared to the United States. And so these data are from a paper by Tom McDade in 2012, and Tom and his team were able to go to lowland Ecuador and do measurements of blood concentrations of C-reactive protein. CRP is a very good biomarker for low-grade inflammation. It's what the American Heart Association uses to determine risk for cardiovascular disease, and levels above 3.0 are considered levels that would confer risk to cardiovascular disease. So all these values that we're looking at here are below that threshold by the American Heart Association. But what you can see is that if individuals came into the clinic and they had no infection, their CRP values are very low, less than 0.5. But if they came in with an infection, the CRP concentrations are almost double, up to 1.0. But if you compare these values in people with infections in lowland Ecuador to the average CRP concentrations in the United States in males and females, overall, those living in modern urban societies like the U.S. tend to have increased CRP concentrations, which is suggesting an exaggerated inflammation under these conditions, albeit a low-grade one. So back to this idea of psychiatric disorders, it's disorders of a failure of immunoregulation. I'd just like to point out that several neurodevelopmental conditions and psychiatric disorders have been found to have reduced regulatory T cells. So again, these are the immune cells that have the capacity to suppress inappropriate levels of inflammation. So in this study, we'd like to see more studies like this. This was a small study showing that children with moderate autism had reduced regulatory T cells measured in the blood, and children with severe autism had dramatically reduced regulatory T cells. And this goes along with a host of studies showing that there's an increase in chronic inflammation in children with autism. Likewise, similar observations have been made with depression and post-traumatic stress disorder. In all of these conditions, the percentage of regulatory T cells circulating in the blood has been found to be reduced. Another argument that would support the idea that there's a lack of immunoregulation in those that are at risk for post-traumatic stress disorder is the finding published in 2015 that individuals with the diagnosis of PTSD have a higher risk of any autoimmune disorder. You can see that at the top of the graph. And they also have higher risk for some specific autoimmune disorders like thyroiditis, inflammatory bowel disease, and rheumatoid arthritis. This is consistent with the idea that there's simply a lack of the ability of the body's own immune system to suppress inappropriate inflammation, in this case, inflammation that's directed against self-antigens, leading to autoimmune disease. Another line of evidence is this study that was published in 2014. This was a group of nearly 2,600 war-deployed Marines, and in this case, they measured CRP, and this is a biomarker of inflammation, at boot camp, and again, post-deployment. And what they found is that, so remember, this is the cutoff for the American Heart Association for cardiovascular disease, 3 milligram per liter. What they found is that individuals that had higher CRP concentrations at boot camp had higher risk for development of PTSD symptoms after deployment. So if in fact inflammation and this lack of immunoregulation is a risk factor for development of PTSD, would it be possible to provide an intervention, restore the immunoregulation, suppress inappropriate levels of inflammation, and prevent the development of a PTSD-like syndrome? To address that question, we used animal models, but before going into that, I want to talk about what these old friends are. So there's 3 general categories of old friends that can suppress inappropriate inflammation. The first is what we would consider the human microbiome. So these are organisms that are part of the co-evolved human microbiome. They include microorganisms like Bacteroides fragilis, Lactobacillus reuteri that you can buy as probiotics. Akkermansia mucinophila is a highly specialized bacterium that eats mucins in the mucus layer lining the gut intestine, and has been associated with driving immunoregulation and suppressing inflammation. Another example, perhaps somewhat surprisingly, is what are called the old infections. So these are infections that were present during the hunter-gatherer period of human evolution, usually harmless, transmitted by the fecal-oral route very early in life, that have been depleted since urbanization. So there's evidence that parasites played this role: hepatitis A virus, Toxoplasma gondii, some strains of T. gondii, Salmonella, and H. pylori. We'll come back to H. pylori later. The third category, which I'll focus on today, is the harmless environmental organisms in mud, untreated water, and fermenting vegetable matter. These are sometimes called pseudocommensals. They include lactobacilli and environmental saprophytes. A saprophyte is a bacterium that lives off of decaying organic matter, and that includes Mycobacterium vaccae, which I'll be talking about and showing you some data later in the talk. In general, our exposure to all of these classes of organisms has been reduced in modern urban societies. So let's talk first about the first category of old friends, the human microbiome. This slide is just to make the point that our microbiome in modern urban societies is thought to be quite different from what we would consider to be a more ancestral microbiome that you can find in hunter-gatherer societies, like the Yanomami Amerindians, Guahibo population, both of these in upper Amazon basin in Venezuela and Colombia, and also the Malawi populations. And so there's a consistent alignment of these hunter-gatherer populations in terms of the community structure of the microbiome. So these are fecal samples, and simply looking at all, say, 500 or 800 different species that are found in the microbiome, looking at these communities, and we'll see a few of these plots later. These are PCOA plots, and what this is showing is that if samples are close to each other, like these two, their microbial communities are very similar. If samples are far apart in PCOA space, these communities are very different. So what this is telling you is that the U.S. Microbiomes from U.S. individuals cluster together, and they're very different in terms of their community structure to hunter-gatherer societies. This is a separate type of metric of the human microbiome. This is bacterial diversity, phylogenetic diversity, or PD. And in general, we think higher diversity. So here on the y-axis scale is a healthier microbiome. And today, the population with the highest alpha diversity of gut microbiome is the Yanomami Indian population in the upper Amazon basin. They have a diet that consists of wild bananas, seasonal fruits, plantains, palm hearts, cassava, birds, small mammals, fish, small fish, crabs, and frogs. So what we know is that stress decreases the diversity of the microbiome, and that provides opportunities for opportunistic pathogens to get a foothold and to cause infection and disease. So that's been established experimentally by Michael Bailey and his colleagues. And so simply looking at alpha diversity, it looks as if those in the United States population have reduced diversity, and this may be a less stable microbiome and perhaps not as resilient as you might find with populations with higher diversity. So I've included my own microbiome report from the American Gut Project. You can see the URL for the American Gut Project down here in the lower right. You can also have your gut microbiome analyzed by the American Gut team, Rob Knight and his colleagues. Simply, they'll send you a kit. You take a swab of stool, and then put that back in a sterile tube and send it back to them. They analyze it, and they send you back a report like this on your own gut microbiome. So you can see here's my sample on the left, and as is typical for Americans, my sample has a relatively equal abundance of what are called firmicutes and bacteroides. And so these are two phyla of bacteria, the most abundant phyla in American populations. And in general, this is in the order of abundance. So firmicutes tend to have the highest abundance. This includes the lactobacilli that you find in probiotics like yogurt, kombucha, and other foods. And then bacteroides is next. Proteobacteria are third. These are of interest because they are gram-negative and contain lipopolysaccharide in their cell wall, which can be potently pro-inflammatory. And then fourth is actinobacteria. This is the phylum to which the environmental saprophytes belong, including mycobacteria, which we'll be talking about later. This is a rare abundance phylum, but we think it may be particularly important, as I'll show you later in the talk. So here's the average relative abundance of different phyla in the American Gut Project sample. Here's comparison with someone with a similar diet, males only in my case, similar BMI, similar age, and Michael Pollan. So Michael Pollan has written and spoken a lot about diet, and I think he's quoted as saying, mostly plants, not too much, kind of summarizes his philosophy. So in my case, in Michael Pollan's case, by far the microbiome is dominated by firmicutes and bacteroides, and not so many proteobacteria. So you also get a report of how you align in PCOA space with other samples in the project. And so in brown are the fecal samples, in blue, dark blue are the oral samples from the mouth, and in dark green are samples from the skin. You can see that this is a fundamental property of the microbiome, that the fecal microbiome is different from the skin microbiome, and that's different from the oral microbiome. This plot shows how, in my case, I compare with other individuals in my country, the United States, and those with a Western diet, Amerindians, which is hard to see on the screen, but they're down here, and also Malawians, which are spread out here in this PCOA plot. And then finally, there's some attention that's been given to the firmicutes to bacteroides ratio, and here it's showing firmicutes abundance, and my sample is somewhere here in the middle with about 50-50, which is pretty typical for modern urban societies. So the American Gut Project now has over 10,000 samples. They've summarized some of their findings here, which again you can find on their website. They have a large number of samples, as I said, over 10,000. Your microbiome changes as you grow. As you get older, your microbiome becomes more diverse. We know that antibiotic usage affects our microbiomes, reduces diversity, leading to a less healthy gut environment. And this is an interesting finding, that the more different types of plants a person eats, the higher their gut microbiome diversity. I think on the survey that they're using, the highest responses, I eat greater than 30 different plants. And this is interesting because we know that plants have their microbiomes. The bacteria that live inside plants are called endophytes. And an interesting piece of trivia is that a 3 to 4 leaf spinach plant has over 800 different species of bacteria inside the plant that you can't wash off. And so just like humans, plants have microbiomes, and when you eat fresh plants, you're eating the microbiomes of those plants. So one approach to increasing diversity of microbiome may be to eat more plants. And also, alcohol consumption also affects microbiome diversity. Those who had at least one drink per week had a more diverse microbiome than those who abstained from alcohol. Okay, so that kind of summarizes the commensal organism. What about these old infections? This is the second category of old friends. And I want to discuss a case study, which is Helicobacter pylori. It's the most common bacterial infection worldwide. It co-evolved with humans, at least since humans migrated out of East Africa 60,000 years ago. And originally, H. pylori came to fame because it was discovered that it's a causal agent in duodenal ulcers, which were very common several decades ago. And in fact, the person who discovered that won the Nobel Prize. It's since been discovered that H. pylori, particularly during development, has potent immunoregulatory effects. So in a sense, you can consider H. pylori an old infection. I'll show you how that works in a moment. So here's the immunology. I don't expect anyone to understand all of these abbreviations here, but just like I showed in an earlier cartoon, the bacteria is binding and interacting with what are called regulatory dendritic cells. This results in the release of anti-inflammatory cytokines like interleukin-10 and TGF-beta, and having interleukin-10 and TGF-beta around tends to bias the differentiation of T cells toward these regulatory T cells that produce anti-inflammatory cytokines. And by biasing toward the production of regulatory T cells, this limits the production of T cells that drive inflammation, like the Th1 cells and Th17 cells that can cause immune disorders. So H. pylori is able to bias the differentiation of T cells toward regulatory T cells, and dendritic cells that have been exposed to H. pylori tend to be poor inducers of these pro-inflammatory immune cells. So overall, we would consider H. pylori, if present during development, have immunoregulatory and anti-inflammatory effects. Here you can see what's happened in terms of H. pylori prevalence. This is showing prevalence of H. pylori infection in various underdeveloped countries compared to the United States. And as you can see, especially during childhood, there's a much higher prevalence of H. pylori infection in these non-U.S. countries. And of particular concern, the exposure among childbearing women is low, suggesting that even women during childbearing years may have low immunoregulatory inputs. So the U.S. population aligns very well with other urbanized countries, including those in Europe and Japan. So having introduced first the commensal microbiome, then the old infections, now I'd like to turn to the third category of old friends, which are these environmental pseudocommensals. They're called pseudocommensals or false commensals because throughout human evolution they would have been present in high numbers, high quantities in the drinking water, for example, but they don't tend to colonize at least the gut mucosa. Recent studies have found that Mycobacterium in particular are highly abundant in the airways, and so the lack of detection of Mycobacterium in the microbiome may simply be a result of the fact that most studies study the gut microbiome as opposed to the microbiome of the airways. So one of these mycobacteria is Mycobacterium vaccae. Vaccae is based on the Latin word for cow, and based on the fact that this species was originally identified in cow dung, but it's an immunoregulatory environmental saprophyte. So normally it lives off decaying organic matter. Mycobacteria tend to be high in soils that have high organic content, so peaty soils, black swamps, that kind of thing. And the history of this particular strain of Mycobacterium vaccae goes all the way back to the 1970s, when John Stanford and his colleagues noticed that in clinical trials for vaccination against leprosy, the efficacy of the vaccine depended on the geography. So in some geographical areas, these vaccines were highly successful. In other geographic areas, they were not very successful at all. And so they went to an area around Lake Kyoga in Uganda, which was an area where these vaccines were highly successful, with the idea of trying to find some environmental factor that could explain why these vaccinations were so highly successful. When they got to the lake, they found that the shores of the lake were lined with this orange slime, and the orange slime turned out to be Mycobacterium vaccae, which is shown here in a scanning electron microscope image. And so people living around the lake would be exposed to high concentrations of mycobacteria, using that for drinking water, for cooking, and as we'll see later on, these bacteria don't have to be living to have an effect on the immune system. So we then wanted to ask the question, if we immunize in our animal models, if we immunize animals with a heat-killed preparation of this Mycobacterium vaccae, which was subsequently found to have these potent immunoregulatory effects and anti-inflammatory effects, can we then prevent development of a PTSD-like syndrome? So after discovering that Mycobacterium vaccae was in fact important in altering the efficacy of these vaccinations against leprosy. This was because Mycobacterium vaccae is closely related to Mycobacterium leprae, and vaccae appears to be acting as an adjuvant. In addition to that, it also has these immunoregulatory properties. So it increases the differentiation of regulatory T cells and production of anti-inflammatory cytokines. So we wanted to test this in a model of chronic stress to see if we could prevent the development of outcomes of chronic stress. And for this, we chose to work with Dr. Stefan Reber, who at the time was at University of Regensburg in Germany, and now is at the University of Ulm in Germany. This is a paper describing the potential use of the experimental model as a model of post-traumatic stress disorder. I won't talk about why we think it's a good model for PTSD, but I've shown you the paper here if you're interested. So the model consists of a large dominant mouse co-housed with 4 subordinate mice. So in nature, mice are social animals, and they spontaneously establish these dominant-subordinate hierarchies, much like primates. And in this particular scenario, the dominant mouse will quickly exert dominance over these smaller mice. Smaller mice show these stereotypical, species stereotypical behavioral responses, including what's called upright defensive posture, where they stand up and freeze. And that's a signal to the dominant mouse that they are in fact subordinate and to leave them alone, and that they're not going to be a threat to the dominant or the dominant status. So the chronic subordinate colony mice, the CSC mice, are stressed in this protocol, and I'll show you in a moment what those stress outcomes are. But this is the general design for our experiments, where we immunize animals once a week for 3 weeks, and then we expose them to this chronic subordinate colony housing model, which is 19 days. And then we do behavioral testing for anxiety and collect tissues for analysis of tissues. So the controls for these studies are single house controls. We use single house controls because mice spontaneously form dominance hierarchies, even if they're all the same size. Okay, so what do we see in this model? We see evidence of changes in the adrenal gland, adrenal hypertrophy. So the adrenal gland grows. This is a common response to stress in mammals. Cells lose their responsiveness to glucocorticoids in vivo and in vitro, and then the sensitivity of immune cells to the stress hormones, glucocorticoids, is also decreased. There's also increase in anxiety as measured by a number of tests, and evidence of some changes in related to somatic disorders, including histological damage to the colon. This is a spontaneous colitis, and so there's inflammation in the colon. There's increased risk for inflammation-related colon cancer, and there's increased risk of severity of chemically induced colitis, which is a model of inflammatory bowel disease. So when we conducted these studies, we injected whole heat-killed Mycobacterium vaccae 3 times, as I mentioned, and then we looked at their behavioral response during the very first hour of exposure to the dominant male. And what we noticed right away is that mice that had been immunized showed a dramatically different behavioral response to this stressor. And specifically, they showed decreased submissive upright postures. So we interpreted this as a shift in their behavioral strategy toward a more proactive response to the stressor. Interestingly, a passive or subordinate response during trauma in humans is also associated with an increased risk for PTSD later on. And so the mice were still subordinate, as indicated here by a negative score, but they were far less subordinate than the control animals. And they were far more likely to show proactive behavioral responses, either chasing or attacking the dominant male in the cage. So we wanted to look at the gut microbiome in the stressed animals and the animals that had been immunized. And what you can see is this alpha diversity. In its simplest form, this can be viewed as a number of different species in the gut microbiome. And you can see that in all groups, the diversity goes down after stress exposure. And this is a very consistent finding with stress exposure in animal models by Michael Bailey and his colleagues and others. And there's some evidence for a protection from the stress-induced decline in diversity of the gut microbiome in individuals that were immunized. If we look at the beta diversity, time is on a fixed axis, so these are the 3 immunizations before the stress starts, and this is day 1 before the stress starts, this is day 8 and day 15, and after stress begins, which is shown here with this gray shading, you can see this kind of expanding universe of beta diversity of gut microbiomes. In other words, what this is saying is that after stress exposure, all the microbiomes of the individual mice are getting different from each other in almost stochastic or unpredictable ways. And so you see this expansion of beta diversity among all the animals that were stressed. And we did find an effective immunization decreasing the stress-induced increase in beta diversity. So we would interpret that as a stabilization of the gut microbiome. So it doesn't prevent changes in the gut microbiome, but it had a small stabilizing effect. Here's another way of looking at what stress, what impact stress had on the microbiome, and you can see that stress on day 8 and 15 among the CSC mice induced a massive increase in proteobacteria. Specifically, this was driven by bacteria that belong to the genus Helicobacter acae, or family Helicobacter acae, genus Helicobacter. And you can see that the immunization in red did not prevent the stress-induced increase in Helicobacter. And notice that this is several orders of magnitude increase in Helicobacter abundance. We think this is very important. We know from other groups that glucocorticoids, the stress hormones like cortisol in humans or corticosteroid in animals, can increase the proliferation of Helicobacter. And this is mediated by the glucocorticoid receptor. So it may well be that simply being exposed to stress induces the proliferation of Helicobacter. In our case, the immunization did not prevent this, but what's interesting is that it prevented the colitis. And so you can see here is a stress-induced colitis, an increase in inflammation in the colon, and this was attenuated or prevented in the individuals that had been immunized with Mycobacterium vaccae. This is a PCOA plot showing that this is before stress, the open symbols. This is after stress, the closed symbols. And these vectors are showing which species are driving this stress-induced shift in the microbiome composition. And you can see it's dominated by Helicobacter species. In contrast, this genus, Mucospirillum, goes down, and Mucospirillum has been suggested in other studies to be a biomarker of gut infection. And so in the presence of gut infection, the mucus layer is disrupted, and consequently Mucospirillum, which feed on mucus, don't have a niche in the gut microbiome, and they disappear. And they only come back after the gut infection resolves. So here is a study looking at this chemically induced colitis. You can see that stress increases chemically induced colitis in this model of inflammatory bowel disease, and this is prevented by immunization with Mycobacterium vaccae. So importantly, stress induces from mesenteric lymph node cells. So these are immune cells that have been isolated from lymph nodes that are draining your gastrointestinal tract. In this case, these immune cells were releasing large amounts of interferon gamma and pro-inflammatory cytokine. And this stress-induced increase was prevented by the immunization. Again, a massive increase in interleukin-6. This was prevented, another pro-inflammatory cytokine. This was prevented by the immunization. And perhaps just as important, instead of producing interleukin-6, animals that were immunized were producing large amounts of interleukin-10. Notice the scale here. These are massive increases in interleukin-10, an anti-inflammatory cytokine. And so the immunization, although it's not preventing changes in the microbiome, it's preventing pro-inflammatory responses that we think are driven by the microbiome. So it's allowing the host to tolerate what we might consider as a microbiome with high potential for inflammation. So just a few more slides, and then we're coming to the end here. We've done a small exploratory study with Sean Hemmings and her colleagues at Stellenbosch University in South Africa. In this study, we were looking at PTSD subjects and trauma-exposed controls in South Africa. And so everyone was exposed to trauma, but as is typical, only a subset of individuals exposed to trauma go on to develop PTSD. And what we found was that individuals that had high relative abundance of actinobacteria and Verruco microbia, which included mainly Akkermansia muciniphila, had a very low risk of PTSD. So in contrast, having low abundance of actinobacteria, which includes Mycobacterium vaccae, for example, was associated with a higher risk of going on to develop PTSD. This may give some evidence of mechanisms involved. This was a study done by Carl and colleagues just recently published, showing that high relative abundance of actinobacteria is associated with low gut permeability in response to a stressor, in this case a 51-kilometer ski march. What about our future directions? Where are we going in the future? I'd like to point out the Showerhead Microbiome Project. We're doing this with Noah Fear here at CU Boulder, Rob Dunn at NC State, Nate Sanders, University of Copenhagen, and Jen Honda and Ed Chan at National Jewish Health. And so we're studying the showerhead because we know that showerheads and municipal water supplies are a major source of mycobacteria. So if you look in soils, mycobacteria abundance is around 1%, but if you look in showers and municipal water supplies, it's about 15% and can be as high as 98%. And so we're looking at hundreds of samples of showerheads across the United States and the rest of the world in order to find out what types of mycobacteria are found in municipal water supplies, and are they correlated with any elements of the water chemistry or other factors that we can identify. And then we're also in a project funded by the Alfred P. Sloan Foundation looking at aerosolized mycobacteria. So in these studies, the bacteria are simply being aerosolized, and we're allowing the mice to breathe mycobacteria. So they're being aerosolized from a water source, and this would be very similar to what you might experience in your own shower, where the shower head is aerosolizing mycobacteria and then you're breathing it in. And in fact, Noah Fear and his colleagues have found that mycobacteria abundance is higher in homes than it is outside the homes, and that it's thought that these mycobacteria are in fact coming from shower heads and other water supplies. So in conclusion, exposure to what we would call immunoregulatory old friends has declined dramatically in developed countries in the last 50 years. Psychiatric disorders are associated with decreased regulatory T cells, decreased immunoregulation, and increased inflammation. We found that immunization with Mycobacterium vaccae prevents stress-induced inflammation and anxiety and fear responses. And this suggests that microbiome-based interventions, sorry about the typo, to increase anti-inflammatory immunoregulatory signaling might be considered for prevention and treatment of psychiatric disorders. So with that, I'll end, and I'll take any questions that attendees of the webinar might have. Thank you very much for your attention. It's been a pleasure.
Chris, thank you very much for the work that you do and are doing, and for an excellent presentation on a complicated topic. One of the questions that a few people have asked is, if there's somebody who does have a psychiatric condition, is there something that they may want to do to affect inflammation, vis-a-vis diet?
Yeah, that's an excellent question, and I would say yes. To the extent that inflammation may be a risk factor, there are ways to including lifestyle choices, to decrease inflammation. So there's a number of anti-inflammatory diet protocols that have been published. You may want to find one of those. A good example is if you eat a hamburger, you have a very rapid inflammatory response. But if you include a slice of avocado on the hamburger, it suppresses that inflammation. So these dietary choices that we make are very important for inflammation. Also, exercise is important, sleep is important. And so these are lifestyle factors, not to mention aspirin or other anti-inflammatory drugs that could have an impact as well.
With regards to potential steps for prevention, what types of things do you see coming in the short term? Where do you see the research bringing us in terms of interventions for prevention?
So clearly, lifestyle factors should be on everyone's mind, because that's something that is accessible to most people if they're informed. And another alternative may be probiotics. We're finding that many probiotics, in other words, live bacteria that confer a health benefit for the host, that you find in yogurt and kombucha, many of these probiotics, as long as they have immunoregulatory and anti-inflammatory effects, may be beneficial. That hasn't been fully demonstrated yet by any means. I would say we're conducting a clinical trial with Lisa Brenner at the VA in Denver, looking at an immunoregulatory probiotic in PTSD subjects with mild traumatic brain injury. So those types of trials are ongoing, and hopefully within a few years we'll have more information about potential impacts on stress resilience and other outcomes.
And above and beyond what you described, is there any work that's looked at different general diets, a vegan diet and other diets that people have that may sort of provide some information about this?
That's an excellent question. I'm not aware of, this is not to say that hasn't been done, I'm simply not aware of studies looking at comparing the diverse types of diets that you've mentioned. I am aware of some studies using some dietary interventions like omega-3 fatty acids, which have shown some promise, and so diets high in these types of fatty acids may in fact be beneficial. But there's a lot more work to be done, and that was in the context of depression. So this is in the context of psychiatric disorders.
Right, right. And you speak about the issue of stress, which we've, in our field we look at in general, but you're really coming at stress from a different direction in terms of the role of stress and inflammation. And I'm just curious where you see future directions of that in future research.
Yeah, I think we're on the verge of a paradigm shift, I think, because when we think in classic terms of stress, we think of activation of the hypothalamic pituitary adrenal axis and glucocorticoid hormones. We think in terms of activation of the sympathetic nervous system, but it's been less well recognized that there's also an inflammatory response to stress. And the potential relevance to psychiatric disorders, I think, is illustrated most clearly by a study by Tad Pace and his colleagues, where they they looked at a social stressor, a Trier social stress test, and then evaluated interleukin-6 response in depressed patients and healthy controls. So remember, IL-6 is one of these pro-inflammatory cytokines. And what they found was that there's a massive increase in interleukin-6 in depressed patients. It's well above that you would find in a healthy control individual. And so most studies that have done meta-analyses of inflammation and depression and schizophrenia, PTSD, and other psychiatric disorders have found this low evidence for low-grade chronic inflammation. But I would suggest that's really just the tip of the iceberg. And what we're missing is that the fact that the whole immune system is lacking immunoregulation, and consequently, when a stressor comes along, there's a hyperactivation of the immune system and a hyperinflammatory response. And we don't often test that in our clinical studies, but I suspect that that's the case.
Well, again, I want to thank you, Chris, for your ongoing work in what may really help change our approach to treatment and prevention in a significant way. Thank you for taking the time to do such an outstanding presentation for us, and look forward to hearing more as you continue the work down the road. So thank you very, very much. I also want to thank our audience for joining us today. All of the research that we fund is made possible through private donations. And if you'd like to make a contribution, please visit our website at bbrfoundation.org or call us at 1-800-829-8289. The webinar has been recorded, and if you've missed any portion of the presentation or would like to share it with a family member or friend, please visit the events page on our website. Finally, I hope people will join us again next month when Dr. Dolores Malaspina will present a webinar entitled Searching for the Seeds of Psychosis. This will take place on Tuesday, August 8th at 2 p.m. Eastern Time. Once again, thank you for joining us, and enjoy the rest of the day. Take care.